Thank you very much. Together with my colleague Adriana Frone, we will chair the section presentations. And we will start with a presentation of our colleagues from the National Patenting Office on the importance of patenting protection of polymeric nanomaterials. Okay, we'll have to ask our foreign guests to be a little bit patient because as this is a very important subject for our young colleagues, the presentation will be Romanian as to not lose anything in translation. And after that, we'll go to the regular oral presentations.
Se poate și fără microfon, cred că mă puteți auzi foarte bine. Mă auziți? Da. Ok, bună ziua tuturor. Numele meu este Ileana Bondar, lucrez la OSIM. Mă simt deosebit de onorată și vreau să mulțumesc pe această cale, în primul rând invitației pe care am primit-o la OSIM de a participa la acest eveniment deosebit de important, atât academic și științific. Mă simt, vă spuneam, onorată personal pentru că mă aflu încă o dată în prezența dumneavoastră, a cercetătorilor și nu în ultimul rând a inventatorilor, celor a căror muncă, îmi place să o spun din tot sufletul, că duc lumea mai departe. Ceea ce mi-am dorit astăzi să vă expun foarte pe scurt, pentru că lumea brevetelor vă este în mod sigur cunoscută, cel puțin măcar o dată ați venit la OSIM să depuneți o cerere de brevet care să încununeze activitatea dumneavoastră de cercetare. Fapt pentru care doream să vă prezint, de fapt, cele două aspecte foarte importante, zic eu, privind brevetabilitatea și anume dezvăluirea invenției și raportul de documentare. Brevetul de invenție, din punct de vedere al dezvăluirii, este un titlu de protecție eliberat de OSIM, care conferă titularului acel monopol de exploatare, acele drepturi exclusive de utilizare, acordat de stat în schimbul dezvăluirii soluției tehnice. Sigur, dezvăluirea este reglementată prin lege, este vorba de legea 64/1991, republicată în 2014, dar și de regulamentul de aplicare al acesteia. Și anume, legea prevede în articolul 17 că invenția trebuie să fie dezvăluită în cererea de brevet suficient de clar și de complet, astfel încât o persoană de specialitate să o poată realiza. Iar regulamentul în articolul 37/2 spune: dezvăluirea clară și completă a invenției va avea în vedere cererea de brevet de invenție considerată în totalitatea sa, aceasta reprezentând descrierea, revendicările și, după caz, desenele. Dezvăluirea, sigur, este foarte importantă. De ce? Pentru că, în primul rând, este cea care creează premizele, condițiile unei documentări, astfel încât să se poată analiza ulterior condițiile de brevetabilitate și respectiv de acordare a unui brevet de invenție. De aceea este practic prima condiție înaintea celor de brevetabilitate să fie analizată atât la nivel național cât și la nivel european. Deci este o tradiție, este, dacă doriți, putem spune, în practica de examinare la nivelul Oficiului European de Brevete, a oficiilor naționale și a Oficiului Român de Brevete privind analiza cererilor depuse în vederea brevetării. Astfel, sigur, această dezvăluire este foarte importantă pentru că dezvăluirea inițială este cea care stabilește limitele protecției ce urmează a fi conferite. E vorba, în special, mă refer la modificările ce pot fi aduse unei descrieri și anume, la data de depozit avem o documentație de brevet constituită din descriere, revendicări și desene. Legea prevede că solicitantul din proprie inițiativă sau ca urmare a observațiilor făcute de OSIM în notificări poate să modifice descrierea inițială, dar cu o condiție, și anume acele modificări să fie admisibile, adică să nu depășească conținutul cererii inițiale. Deci este foarte importantă dezvăluirea inițială. Avem niște limite, nu le putem depăși ulterior. Chiar dacă OSIM-ul face observații în acest sens, modificările trebuie să respecte limitele impuse de cerere, de conținutul cererii la data de depozit. Atunci când ați depus toate documentațiile, cererea de brevet. Când spun cererea de brevet, nu mă refer la formularul de cerere, ci la toată documentația, adică privind lucrurile în examinarea de fond. E vorba de descriere, revendicări și desene. Sigur, când ar trebui să depunem o cerere de brevet și mai ales când trebuie făcută această dezvăluire? Foarte important. Deci trebuie să se facă înainte de divulgarea prin publicare a rezultatelor cercetării și anume articole, cărți, pliante promoționale, postere, simpozioane, cum este cel pe care îl trăim astăzi. Înainte de folosirea sau comercializarea obiectului invenției și înainte ca prin orice alt mijloc rezultatele cercetării dumneavoastră să fie devenit accesibile publicului. Altfel spus, foarte simplu, întâi venim la OSIM și depunem o cerere de brevet, după care facem public în orice altă conjunctură rezultatele cercetării. De ce? O să mă întrebați. De ce așa și nu invers? Pentru că toate documentele care sunt anterioare cererii de brevet se opun pe noutate. Dacă eu mă duc întâi și fac un poster pe care îl expun la un simpozion sau la o conferință și acolo trec toate rezultatele activității mele deduse din activitatea de cercetare, după care vin la OSIM, la OSIM se face, bineînțeles, o analiză, o cercetare documentară, se analizează în primul rând din punct de vedere al dezvăluirii inițiale și apoi al condițiilor de brevetabilitate și mi se va opune exact posterul meu. Stau și mă întreb, ce argumente am putea să mai aducem dacă am dezvăluit într-un poster înainte absolut toate datele dintr-o ulterioară cerere de brevet. Fapt pentru care recomandarea mea, și nu numai a mea, a celor care lucrează efectiv dosarele de brevete de invenție ale cererilor de brevete în vederea acordării brevetelor, este următoarea. Întâi faceți un depozit național reglementar la OSIM, depuneți cererea cu toate documentațiile, cu o dezvăluire cât mai amplă a invenției, după care o publicați acolo unde aveți ocazia, mai ales în revistele cu foarte bună cotație internațională. Dezvăluirea, sigur, vă spuneam, este foarte importantă cu privire la limitele pe care ea le trasează din punct de vedere al domeniului de utilizare a invenției, astfel încât modificările ulterioare să poată să fie admisibile. Obiectul dezvăluirii este însă și invenția, iar locul dezvăluirii este, de fapt, reprezentat de descriere, desene și revendicări. Și anume, unde putem găsi în cadrul invenției, unde putem face această dezvăluire? Recomandarea mea este să parcurgeți prevederile regulamentului în articolul 16, care tratează sau prezintă foarte, foarte clar toate capitolele pe care trebuie să le cuprindă descrierea dumneavoastră, astfel încât să avem o dezvăluire clară și completă a invenției. Singurul este titlul în care acesta este clar prevăzut, nu trebuie să dezvăluie cu nimic invenția, în schimb, domeniul tehnic, stadiul anterior al tehnicii reprezentat de cel puțin o soluție din stadiul tehnicii cunoscută, apropiată de soluția pe care dumneavoastră doriți să o brevetați, cu dezavantaje în măsura în care le cunoașteți. Problema tehnică este foarte importantă pentru că o invenție este o soluție tehnică la o problemă tehnică, iar problema tehnică totodată ajută foarte mult la analiza activității inventive a invenției dumneavoastră. Expunerea invenției, în care trebuie să apară exact așa cum este invenția revendicată, cu foarte multe detalii. Repet, deci prezentarea soluției tehnice care duce la rezolvarea problemei. Expunerea invenției trebuie să consta cel puțin dintr-un exemplu concret de realizare a invenției. Dacă aveți mai multe variante puneți un exemplu pentru fiecare variantă în parte. Dacă aveți mai multe obiecte, de exemplu, o compoziție, un produs nou, un procedeu de obținere a acestora sau o instalație sau dispozitiv special conceput pentru aplicarea procedeului, atunci trebuie câte un exemplu pentru fiecare dintre aceste obiecte. Prezentarea pe scurt a desenelor și nu numai, deci pe lângă exemplele concrete. Să vă spun de ce este foarte important capitolul desene. În general, ca orice inginer chimist, atunci când este vorba de o instalație, nu citesc dezvăluirea propriu-zisă a invenției, cât mă duc imediat la desene. Să văd, e vorba de o instalație, este reprezentată în mod clar, caracteristic, însoțită de repere, pe care ulterior se le regăsesc în textul descrierii. De multe ori se produc niște mici inadvertențe, astfel încât soluția nu este perfect dezvăluită sau nu este suficient dezvăluită. Examinatorul notifică acest fapt solicitantului, îi se dă un termen de răspuns, termen în care se așteaptă modificările și completările. Repet, este foarte importantă dezvăluirea inițială pentru că acele modificări, răspunsul dumneavoastră și argumentele prezentate să se încadreze în limitele depozitului inițial. Cum spunem noi, conținutul cererii la data de depozit. Revendicările, sigur, reprezintă inima oricărui brevet, pentru că ele sunt cele care determină întinderea protecției conferite de brevetul de invenție. Între revendicări, sigur, și descriere există totdeauna o legătură indisolubilă. Revendicările, legea prevede că acestea trebuie să fie clare, concise și bazate pe descriere. Vă spuneam, legătura dintre revendicări și descriere subliniază încă o dată faptul că acele obiecte au fost dezvăluite de la început în cererea de brevet a solicitantului. Deci e vorba de protecția solicitată pentru acele obiecte din revendicări pe care le-a descris la data de depozit solicitantul cererii de brevet. Vă spuneam că această dezvăluire în desene este importantă, este o practică întâlnită atât la nivel național cât și la nivel european. Desenele trebuie să prezinte, vă spuneam, elementele componente ale unui dispozitiv sau ale unei instalații, așa cum ele sunt revendicate și dacă în descriere poate mai scapă câteodată o anumită precizare sau un anumit detaliu, dar acesta există în revendicări și există în desene, înseamnă că invenția este perfect dezvăluită sau complet sau suficient dezvăluită. Repet, deci toate elementele componente ale unei documentații de brevet sunt luate în analiză încetul cu încetul, pagină cu pagină și vă rog să mă credeți că după 30 și ceva de ani de activitate din dosare, asupra dosarelor aplicându-mă, acestea sunt constatări din munca mea de zi cu zi. Dezvăluirea suficientă. Deci, de multe ori, solicitanții noștri se tem să dezvăluie complet invenția, să ascundă de frica să nu fie copiată cumva, să prevină copierea invenției. Totuși, dacă persoana de specialitate care trebuie să realizeze invenția constată că lipsește un segment, să zicem, dintr-un procedeu, o etapă, ceva anume, care nu i-ar permite persoanei respective să realizeze invenția, se consideră că invenția este insuficient dezvăluită. Deci este foarte important, încă de la început, dezvăluirea invenției să se facă cât mai detaliat posibil. O invenție este clar și complet dezvăluită, sigur, dacă permite terților să înțeleagă contribuția pe care o are invenția respectivă la stadiul cunoscut al tehnicii. Ce pot să vă spun? Ca pe o filozofie, dacă se poate spune, trebuie avut în vedere următorul aspect. Nu poate fi protejat decât ceea ce este revendicat și nu poate fi revendicat decât ceea ce este descris, adică dezvăluit. Lipsa de dezvăluire, sigur, atrage o sancțiune prevăzută de lege și anume, dacă invenția nu este suficient dezvăluită sau nu este deloc dezvăluită, cererea de brevet de invenție se respinge pe acest motiv, lipsa de dezvăluire sau insuficienta dezvăluire. Mai mult decât atât, un brevet de invenție acordat terții au posibilitatea prin lege să formuleze o cerere de revocare pe baza, invocând acest motiv, lipsa de dezvăluire, și am avut un astfel de caz în domeniul fizicii, care s-a judecat la noi, la Comisia de Apel, s-a invocat lipsa de dezvăluire demonstrată și probată de către terți, solicitantul revocator, căruia în final i s-a dat dreptate, în sensul că s-a obținut, brevetul a rămas în picioare, dar parțial, deci a fost parțial revocat. El poate fi și total revocat. Mai mult decât atât, brevetele acordate pot fi și anulate la instanță pe același motiv, lipsa de dezvăluire. Iată de ce insist și recomand: dezvăluiți cu cât mai multe elemente tehnice, caracteristici tehnice, așa zise, prevăzute de lege ca fiind esențiale, adică cele care contribuie la rezolvarea problemei tehnice. Lucrurile par complicate, dar în esență nu. Munca dumneavoastră stă în spate, iar redactarea unei cereri de brevet de invenție, astfel încât din start să fie perfect dezvăluită, suficient dezvăluită, poate fi evident îndrumată, ajutată de consilierii în proprietate industrială. Din nou vă recomand, accesați www.osim.ro, pagina noastră de website-ul nostru, unde există o listă a consilierilor care sunt acreditați de OSIM, a celor care au parcurs cursurile și au dobândit, în urma unui examen, calitatea de consilier în proprietate industrială. Este foarte important și cu atât mai mult din rândurile dumneavoastră ale cercetătorilor. Urmăriți momentul în care OSIM-ul publică organizarea unor astfel de cursuri, participați la cursuri, înscrieți-vă la examenul de consiliere în proprietate industrială, că vă va ajuta foarte mult în redactarea cererilor de brevet, astfel încât să aveți toate eforturile răsplătite și monopolul de exploatare asigurat în brevete cât mai solide. Nu vreau decât să vă dau două exemple foarte simple. Am găsit două brevete recente care au două tipuri de obiecte. E vorba de agregate de argint, adică prin prisma legii acestea reprezintă un produs, caracterizate prin aceea că este sintagma care în redactarea revendicărilor desparte preambulul revendicării de partea ce urmează, și anume de partea caracteristică, cu alte cuvinte, elementele noi care există în partea caracteristică de preambul, cele cunoscute, și doream să parcurgeți ușor aceste rânduri și să vedeți câte detalii tehnice, cu alte cuvinte, caracteristici tehnice esențiale sunt prezentate într-o revendicare independentă de produs, în speță agregatele de argint, astfel încât să fie satisfăcută, îndeplinită condiția dezvăluirii invenției. Da? În același brevet mai exista o categorie de revendicări, e vorba de procedeul de obținere a agregatelor de la revendicarea 1, deci este procedeu special conceput pentru realizarea acestor agregate de argint. Din nou, multitudinea de parametri. Deci un procedeu, conform legii, trebuie și regulamentului de aplicare, trebuie descris prin etapele de procedeu cu toți parametrii în ordinea logică de desfășurare a procedeului. În mod similar, mai avem un exemplu, tot așa, nanoparticule de argint. Am mers pe argint, pe linia aceasta a argintului, în care, în mod similar, deci avem o revendicare de produs, și anume nanoparticule de argint cu dispersie, dar și uitați-vă câte elemente tehnice, caracteristici tehnice esențiale sunt menționate. Ele sunt cele care determină întinderea protecției. Repet, revendicările trebuind să îndeplinească, să fie clare, concise și bazate pe descriere. Deci nu numai elementele noi sunt trecute în revendicări, acestea au un ecou, se regăsesc în descriere și în exemplul concret de realizare a invenției. Și în mod similar, deci al doilea brevet, care cuprinde și o revendicare de procedeu, deci întinderea protecției este asigurată atât pentru produs, cât și pentru procedeul de obținere a acestor nanoparticule de argint, cu revendicările de rigoare. Deci, înainte de a trece la partea a doua a expunerii mele, pot să vă fac aceste recomandări. Când trebuie să dezvăluim invenția, totdeauna înainte de publicare în reviste de specialitate sau cotate internațional sau evenimente cum este cel de astăzi, descrierea invenției trebuie să fie clară și detaliată, să cuprindă toate capitolele așa cum prevede regulamentul, încă de la data depozitului național reglementar, adică din acea zi măreață în care doriți, depuneți cererea de brevet cu toată documentația la OSIM, iar revendicările, inima brevetului, cele care determină întinderea protecției conferite de un eventual brevet, trebuie să fie clare, concise și bazate pe descriere. Partea a doua a expunerii mele va fi și mai scurtă decât prima, poate că prima a cuprins o multitudine de aspecte, dar, repet, izvorâtă din practica mea, din activitatea mea de zi cu zi pe dosare. Raportul de documentare. Raportul acesta de documentare îl efectuează OSIM numai la cererea solicitantului. Este prevăzut, legiferat prin articolul 23 din lege și articolul 40 aliniatul 2 din regulament. Deci, acesta se poate face la cererea solicitantului, cu condiția, evident, a achitării taxei legale de procedură, adică de cercetare documentară. Se face o cercetare documentară care este solicitată fie până odată cu depunerea cererii, fie până în a treia lună de la data de depozit și OSIM are datoria să vă răspundă acestei solicitări de efectuare a raportului de documentare până în a 10-a lună sau până în a 20-a lună, în funcție de data la care solicitați. Recomandarea este cu cât mai repede, cu atât mai bine. Deci, odată cu depunerea cererii solicitați cu condiția achitării și taxei legale, solicitați efectuarea raportului de cercetare sau și de documentare. Ce rost are acest raport de cercetare documentară? Este cel care stabilește stadiul tehnicii, cu alte cuvinte, se face o cercetare în multe colecții, atât de brevete, cât și în literatura non-brevet, a tuturor soluțiilor tehnice foarte apropiate de soluția pentru care dumneavoastră ați depus cererea de brevet. Se selectează, deci, aceste documente și se specifică într-un formular tipizat incidența pe care o au acele documente selectate asupra revendicărilor din cererea dumneavoastră. Sigur, furnizează solicitantului și terților, după publicarea acesteia, informații despre stadiul tehnicii, da, relevant față de invenția din revendicări. Deci, vă spuneam, incidența documentelor selectate asupra revendicărilor din cererea dumneavoastră. Acest raport de cercetare documentară poate să fie însoțit sau nu de o opinie scrisă privind brevetabilitatea. Aici doream să ajung, pentru că reprezintă partea cea mai importantă a acestui raport de documentare, pentru că dacă raportul simplu are doar documentele selectate și incidența asupra revendicărilor, opinia scrisă cuprinde analiză în ceea ce privește unitatea invenției, dezvăluirea invenției și, nu în ultimul rând, îndeplinirea criteriilor, condițiilor de brevetabilitate, noutate, activitate inventivă și aplicabilitate industrială, astfel încât neîndeplinirea uneia dintre condiții este notificată. Deci acest raport de cercetare documentare se publică. Se publică numai el, nu și opinia scrisă asupra cererii în cauză. Ea este comunicată solicitantului, este o notă de confidențialitate pe care o are această opinie scrisă a examinatorului și în termenul acordat de OSIM, solicitantul trebuie să răspundă observațiilor astfel încât să poată să aibă succes cererea formulată și depusă la OSIM. Deci, avantajele pe care le prezintă, vă spuneam, deci în primul rând, el înainte de publicarea cererii are loc realizarea raportului de cercetare documentare. Acesta se poate publica fie odată cu cererea, fie ulterior, dar la o distanță foarte, foarte mică, oferind posibilitatea solicitantului de a decide. Continuă cu achitarea taxelor ulterioare până la eliberarea brevetului sau nu? Pot să vă spun că în prezent am pe masă, în cadrul Comisiei de Apel, a Comisiei de Contestații din cadrul Direcției Juridice în care îmi desfășur activitatea, o contestație formulată împotriva unei hotărâri de respingere. Pe ce motiv credeți dumneavoastră? Pe motivul noutății. O cerere de brevet care a fost întâi, din păcate, tipărită, publicată într-o revistă de specialitate internațională, după care, la ceva timp, o lună și ceva, a fost depusă la OSIM cererea de brevet. Evident, s-a făcut raportul de cercetare, a fost solicitat raportul de cercetare documentară și s-a depistat imediat revista internațională cotată foarte bine, deci internațional, a fost selectată, a fost identificat conținutul cererii. 1 la 1 ca identitate față de conținutul acelei publicații, publicația fiind anterioară, s-a opus propriei cereri de brevet a solicitantului și, evident, cererea a fost respinsă pe motivul lipsei noutății. Deci 1 la 1 identitate între cerere și publicația anterioară. Stau și mă întreb, sigur, solicitantul are dreptul, conform legii, să formuleze o contestație împotriva hotărârii de respingere, hotărâre care este acum soluționată de Comisia de Reexaminare unde eu lucrez. Și stau și mă întreb, ce argumente ar putea să mai aducă solicitantul din moment ce a publicat tot întâi în acea revistă față de conținutul cererii la data de depozit, atâta vreme cât au fost identificate elementele de noutate 1 la 1. Deci, practic, publicația internațională este considerat document distrugător de noutate pentru cererea de brevet. Deci, iată de ce este foarte important acest raport de cercetare documentară. Sigur, solicitantul este în deplină cunoaștere a condițiilor și faptul că s-a depistat acest document public și este distrugător de noutate, nu mai are rost să continue, pentru că lipsa de noutate este prima condiție din condițiile de brevetabilitate ce se analizează privind eliberarea unui brevet de invenție. Dacă nu este nouă, celelalte condiții nici măcar nu se mai analizează. Deci este foarte important de știut, astfel încât acest raport de cercetare documentare aduce la cunoștința solicitantului ce șanse de succes are. Continuă cu achitarea celorlalte taxe, continuă să obțină totuși un brevet, luptă mai departe, dar nu prea are argumente, sau o ia de la capăt, dar cu o altă soluție spre brevetare. Opinia scrisă, vă spuneam, deci cuprinde atât observații privind îndeplinirea condițiilor de dezvăluire, observații precise cu privire la noutate, activitate inventivă și aplicabilitate industrială și explicații privind relevanța documentelor citate în raport asupra revendicărilor corespunzătoare. Recomandarea mea, în urma celor ce v-am spus, este următoarea. Deci se depune cererea de brevet și odată cu ea, sau la diferență foarte mică de timp, să fie cu condiția achitării taxelor legale, să fie solicitat raportul de documentare cu opinie scrisă, care vine atât în sprijinul solicitantului să-l orienteze cu privire la demersurile ulterioare, cât și a terților, pentru că îi informează de existența unei cereri de brevet și a unui potențial brevet ce urmează a fi eliberat de către OSIM pe tema respectivă în condițiile respectării și îndeplinirii tuturor condițiilor legale, astfel încât se evită cercetările paralele și totuși se stimulează creativitatea. Deci, acestea sunt ca și concluzii finale. Avem nevoie de o dezvăluire clară și completă a invenției, avem nevoie de o redactare corectă, clară și concisă a revendicărilor bazate pe descrierea de brevet și nu numai atât, solicităm un raport de cercetare documentară, achităm taxele legale de procedură, astfel încât să reducem considerabil timpul de examinare a unei cereri. Pentru că solicitând acest raport de cercetare documentară, este posibil ca înainte de publicarea cererii, adică până în 18 luni, solicitantul să aibă deja notificarea în care are toate observațiile menționate și să-și poată prezenta punctul de vedere, argumentele sau reformulările ținând cont de prevederile legale. Și atunci, sigur, vom avea o cale de acces mult mai rapidă spre brevetele de invenție.
Vă mulțumim foarte mult pentru prezentare, care, într-adevăr, cred că pentru noi, mai ales pentru colegii noștri mai tineri, reprezintă un subiect foarte interesant. Înainte de a solicita întrebări din sală, aș vrea să apreciez în mod deosebit faptul că printre exemplele pe care le-ați acordat de bune practici sunt și brevete ale colegilor noștri prezenți în sală. Am recunoscut rezultatele voastre și activitatea voastră. Bun, acum dacă vrea cineva să pună întrebări.
Bună ziua, Ioana Ipicea. Doresc să întreb dacă am avut un brevet sau dacă mă uităm când facem descrierea, nu când facem descrierea, deci dacă eu descriu în exemplu ceva și în revendicare uit să pun o parte, e grav? Adică în exemplu am pus tot și la revendicare uit să pun un amănunt.
Sigur, în primul rând, examinatorul vă va notifica părțile lipsă, da. Dacă ele sunt importante, dar dacă sunt susținute și dezvăluite în depozitul inițial, da? Da. Cu delicatețe și cu perspicacitate, vedem cum pot fi reformulate revendicările, astfel încât să nu se depășească depozitul și să poată fi inclusă ceea ce considerați dumneavoastră că ați omis, că nu ați precizat. Dar, repet, revendicările clare, concise și bazate pe descriere. Deci, sigur, nu o multitudine foarte mare de parametri, de caracteristici tehnice ale invenției să fi fost lipsă. Nu. Dar, sigur, prin dialog și țineți legătura cu examinatorul, astfel încât, bazându-se pe dezvăluirea din exemplu, ele pot fi la o adică, dar trebuie analizat foarte bine textul. În general, lucrurile invers se petrec. Există în revendicări și s-au ratat, să zic așa, din cuprinsul descrierii.
Așa. O altă întrebare. Data de depozit, adică când depuneam, aceea este importantă, nu data când apare în bobii.
Nu, evident. Okay. Deci, borna temporală, cum îmi place mie să o numesc, este data de depozit, data la care ați întrunit minime condiții prevăzute de lege pentru acordarea acelei date de depozit, numărul de cerere cu data respectivă. Este borna temporală față de care se apreciază atât dezvăluirea, cât și condițiile de brevetabilitate. Noutate, inventive step, activitatea inventivă și aplicabilitatea industrială.
Mă scuzați, aș vrea aici să fac o nuanță. Până când nu se publică în BOPI, nu sunteți protejați. Clar? Că asta nu vă spune. Deci, publicarea în BOPI înseamnă protecție. Până când nu se publică în BOPI, nu există protecție. Bun.
Dacă îmi permiteți un mic amendament.
Stăm să discutăm.
Publicarea în BOPI este o protecție provizorie pe care
e o protecție provizorie pe care
emiterea hotărârii Comisiei de Examinare.
Până la emiterea hotărârii. Iertați-mă, dar dacă se ajunge dintr-o instanță românească pe o fază de genul ăsta, haideți să nu mai discutăm. Da, bun, deci în momentul până când nu ți apare, n-are ce să fie protejată. Că oricine va invoca în justiție că nu știa. Bun, că n-avea de unde să știe.
Acesta este rolul publicării, pentru ca terții să aibă în vedere că există o cerere depusă și un potențial brevet care urmează, deci, a fi acordat. Iar protecția este o protecție
limitată,
provizorie care este emisă prin publicarea cererii de brevet în buletinul nostru oficial de proprietate industrială.
Deci, ca unul dintre utilizatori, aici mă discut ca unul dintre utilizatori și vă spun, apropo de raportul de documentare cu opinie scrisă, care ar fi interesul la amândouă părțile. Dacă nu reveniți la vechea formă, decizia pe care o vom lua toți cei care știm este să mergem pe VIPO și să vă plătim cât mai puțin dumneavoastră. Pentru că diferența între 6 luni și cele câteva luni care sunt în plus la VIPO, în care obținem același rezultat, este compensată de protecția extinsă la nivel global. Deci, Credeți-mă, 6 luni este prea mult. Eu înțeleg situația, am vorbit și cu colegele, știu problemele de resurse umane cu care toți ne confruntăm, numai că în anumite situații noi avem nevoie de raportul de documentare cu opinie scrisă ieri. Pierdem oportunități de licențiere și așa mai departe. Și atunci, dacă gândim din timp toate celelalte, ne este mai rentabil să facem VIPO pe CET.
Deci, sigur, este interesul dumneavoastră de a depune cererile la WIPO, adică Organizația Mondială a Proprietății Industriale cu sediul la Geneva, OMPI, cum se mai numește.
Da.
Ok, aceștia nu acordă brevet, este
Da,
vorba
e
doar de
cerere,
publicarea și efectuarea raporturilor de cercetare, documentare și publicarea acestora, da, dar nu obțineți brevet. Brevetul urmează mai departe.
Trebuie luat pe fiecare jurisdicție în parte.
Obținut pe cale națională sau pe calea europeană unică, la Oficiul European de Brevete, sub imperiul achitării unor alte taxe europene comparativ cu cele ale oficiului nostru. Ca să fie răspunsul complet. Ok, vă dau dreptate și...
Acum, ca un comentariu, diferența între cele două aspecte, între data și data depunerii, este că tot ce se după data depunerii nu mai sunt opozabile propriei invenții. Dar când vorbim de licențiere, e altă poveste. Este asta ca să înțelegem de ce sunt două opinii. Bineînțeles, bineînțeles. Dar te duci în urmă.
Și pentru faptul că există brevetul. Brevetul este instrumentul de apărare în instanță atunci când există o contrafacere, o încălcare de drepturi.
Și este absolut corect că pentru a ști trebuie să fie public și la momentul ai o bucată. Acum, institutul nostru, ca o paranteză, beneficie și de un foarte bun consilier în proprietate industrială. Cred că și dumneavoastră știți, este unul dintre liderii în cereri de brevete depuse la nivel național. Dacă mai sunt întrebări, în primul rând, scuze. Stai puțin ca să termin și eu. Stai să termin și eu. Bun. Acum, întrebarea care e pe buzele tuturor e de ce durează atât de mult?
Da. În primul rând, avem o zestre de dosare moștenită. 2. Din punct de vedere personal, suntem cam la jumătate din capacitate per global, la nivel de instituție, da? Și ce pot să vă spun, în condițiile în care și solicitanții ne-ar ajuta cu solicitarea acestui raport de cercetare documentară odată cu depunerea cererii sau cât mai repede posibil, evident cu condiția achitării taxelor legale, astfel încât până în 18 luni acesta să aibă o primă notificare de fond. Este foarte important. Sigur că am reduce foarte mult timpul de examinare până la emiterea unei decizii, unei hotărâri de către Comisia de Examinare de Fond.
Foarte scurt, în primul rând, mulțumesc, excelentă și discuția dinainte și prezentarea dumneavoastră, nuanțe fine, chiar am aflat. Întrebarea foarte scurtă, de ce trebuie neapărat să fie internațională publicația? Dacă e românească, nu e tot o dezvăluire și se poate opune?
E vorba de faptul că cercetarea documentară se face la nivel mondial. Da?
Nu, a fost doar un exemplu. A fost doar un exemplu, dacă o publici în România...
Noutatea trebuie să fie absolut. Știu.
Noutatea este mondială. Cum?
Bun, în momentul ăsta... Ne scuzați. Cred că toată lumea a înțeles deja.
Ai dezvăluit în ceva doar românesc.
Bineînțeles că este dezvăluit și este aceeași. Este opozabil.
Da, este opozabil. Absolut, absolut.
Cred că undeva a fost o eroare de comunicare.
În ceea ce privește noutatea.
Mulțumesc. Mulțumim foarte mult.
Mondial.
Sperăm ca și dumneavoastră, ca și noi toți ceilalți să trecem peste probleme de resurse umane, pentru că, într-adevăr, este o perioadă destul de lungă și pentru că, acum, opinia mea, corect este să licențiezi brevete, nu cereri de brevet, chiar dacă ai opinia de scris. Bun, și atunci este posibil să treacă un timp suficient de mare încât să fie depășit tehnic, chiar dacă soluția ta e bună. Vă mulțumim foarte mult. Dacă mai sunt întrebări din sală sau din online. Mulțumim foarte mult pentru prezență și vă așteptăm și la celelalte manifestări organizate.
Când vă stăm la dispoziție, am lăsat în final și adresele de mail la care ne puteți contacta. Suntem oficiu de acordări de brevete și nu de respingeri, de aceea doresc din tot sufletul să avem cereri de brevete cât mai bine redactate, detaliate și documentate.
Thank you again. We'll continue the oral presentations with Iulia Ștefania Voicu with the talk entitled Encapsulation of Polyphenolic Extracts from Vaccinium myrtillus in Functionalized Mesoporous Silica with Anti-Inflammatory Activity.
Hello everyone, today I'm going to present our study concerning the encapsulation of polyphenolic extracts from Vaccinum myrtillus in functionalized mesoporous silica with anti-inflammatory activity. Here are the principal topics that are covered in my presentation, and I'll jump straight to the first subject, the aim of our work. The aim of our work was to evaluate the properties of bilberry fruit extracts free and encapsulated in mesoporous silica nanoparticles, pristine or functionalized with organic moieties. But why those polyphenolic extracts? Why? What do they contain? Natural polyphenols are bioactive molecules that are found mainly in plants. They divide in two classes, flavonoids and non-flavonoids. Anthocyanins belong to the flavonoids class and exhibit some interesting anti-inflammatory and antioxidant properties. Here is the general structure of anthocyanins, and some studies suggest that the highest contents among berries, the highest contents of anthocyanins are contained in bilberries. However, anthocyanins are prone to degradation in air, and their bioavailability is low due to their low solubility. To overcome those drawbacks, we chose to encapsulate those extracts in mesoporous matrices, especially mesoporous silica-based supports, due to their interesting properties like high pore volume, large specific surface area, good biocompatibility, chemical stability, and the possibility to tailor their surface. Two polyphenolic extracts were obtained in ethanolic solution acidified with citric acid, following two different routes. The first one in a conventional four stages of 1 hour each extraction at reflux with solvent replacement, and another one in inert atmosphere in 1 stage of 3 hours at the same temperature as the first. The chemical profiles of the polyphenolic extracts was assigned using spectrophotometric methods and HPLC PDA chromatography. The spectrophotometric methods helped us to reveal the total contents of polyphenols, flavonoids, and anthocyanins, as well as the radical scavenging activity correlated with the antioxidant properties. The highest contents of anthocyanins were assigned for the conventional bilberry extract, whereas the highest contents of flavonoids was discovered to be for the inert atmosphere extract. The radical scavenging activity was best assigned with DPPH assays for both extracts. Up to 8 different compounds were identified in the extracts, the most abundant being delphinidine chloride, rutin hydrate, and chlorogenic acid. We proposed 4 different mesoporous matrices to encapsulate the extracts, the first one being pristine MCM41 silica, another one a cyanoderivate being prepared by post-synthesis method that was subsequently hydrolyzed in acidic medium, resulting in the carboxylic acid derivate. A third one, a mercaptoderivate synthesis by co-condensation approach, and a fourth one consisting of the mercaptoderivate nanoparticles coated in fucoidan. Those supports were characterized using different techniques like small angle X-ray diffraction. The patterns suggested that all the samples were crystalline, lower degrees of crystallinity being obtained for the functionalized supports. You can also note the 100 Bragg refraction that is present for all the samples. The FTIR spectra for pristine and functionalized silica revealed, besides the bands specific for the silica matrix, the bands specific for the organic moieties, of course, for the functionalized ones. You can note the specific vibration of the cyano group, the carbonyl group, and others. The nitrogen absorption desorption isotherms recorded at 77 Kelvin were all type 4 isotherms characteristic for mesoporous materials. You can also note the textural parameters that are high, specific for those kind of materials, but somehow lower for the functionalized ones due to the presence of the organic moieties on the inner walls of the pores. The CEM investigations revealed that all the samples possess spherical particles with diameters lower than 100 nanometers. You can see that the fucoidan coated particles tended to form agglomerates. The extracts were loaded on the silica matrix using the impregnation method, followed by solvent evaporation in high vacuum. The extract contents were determined using the thermogravimetric analysis, and due to the high porosity of the materials, high contents of polyphenolic extracts could be encapsulated up to 39%, as you can see. The biological evaluation was assessed using mouse macrophage cell line RAW264.7. The biocompatibility was assessed, and the other analyses were assessed using two concentrations, 10 micrograms per milliliter and 100 micrograms per milliliter. For the biocompatibility assessment, all the samples possess almost no cytotoxicity, but one, the fucoidan-coated nanoparticles showed a slight decrease in cell viability, but it can be considered safe. The anti-inflammatory activity assay was a nitrogen monoxide assay. The best candidate for this kind of analysis was the carboxylic acid derivate material because it showed the slightest, not exactly the slightest reduction in NO production, but coupled with the cell viability, it exhibited the best properties. The cyclooxygenase inhibition assay showed a good selectivity for COX2 over COX1. And in conclusion, we obtained two polyphenolic extracts that we chose to encapsulate in 4 different mesoporous silica-based supports. They showed good anti-inflammatory and antioxidant properties, and they showed almost no cytotoxicity. And they can be used in future biomedical applications. Thank you for your attention.
Thank you for this nice talk. And there are questions in the room or online?
Why the need to encapsulate this extract? Because the route of administration of such products is usually the oral way.
Some of them are prone to degradation in the gastrointestinal tract and to survive in the body, to say that. And of course, for the storage.
Yes, that are good reasons for that. Thank you.
Thank you again. We move to the next one.
Thank you very much. Thank you. Our next presentation, it's a cold active aldehyde dehydrogenase from Flavobacterium as a new enzymatic label for DNA aptamers, a preliminary studies. Authors Andrei Aftodiev, Georgiana Necula, Petrăreanu, Cristina Purcărea, Camelia Bale, and Alina Vasilescu.
Good afternoon. My name is Todiev Andrea, and today I'll present you a co-active aldehyde dehydrogenase from Flavobacterium as a new enzymatic label for DNA aptamers. So firstly, what is an aptamer? So aptamers are short single-stranded DNA or RNA molecules that can selectively bind to a specific target. Aptamers have been generated against cancer cells, bacteria, and viruses, and they also use in the fight with coronaviruses. A few things about the aldehyde dehydrogenase, which is shortened as FALDH. So it's an enzyme that has a histidine tag attached to the N-terminal end, and that histidine tag has an affinity for the nickel nitriloacetic complex. It's also an aldehyde dehydrogenase with proven potentials with biosensors, being cold active recombinant enzymes which functions in the wide temperature range. Here you can see the scheme of my experiments. So we started from the aptamer with the thiol end group that was attached to nitriloacetic acid through maleimide, and we obtained that complex that you can see there. And after that, that complex, as I said previously, FALDH has an affinity for nitriloacetic acid. So we attach that complex to FALDH in the presence of nickel chloride. And we obtained our product of interest. This was our main reactions. But in parallel, we also made the same reactions, but instead of using nickel chloride, we used EDCNHS to see how our complex will behave if we would have a covalent coupling, not a non-covalent one. So mainly it's pretty easily to write these reactions and to mix all these compounds together. But we also needed to see if our complex was truly formed. So firstly, we use UV spectrometry analysis. Here you can see two figures in which in both of the figures, there are present the spectra of the enzymes FALDH and of the aptamer, we used. In the first spectra, you can see, in the first figure, you can see the spectra of the aptamer of the complex in which we use the nickel chloride, and in the second figure you can see the complex in which we use EDCNHS. So as I said in the first slide, aptamers contain DNA. So foreseeing that our complex is truly has aptamers in them, we need to see a peak at 260 nanometers. And as you can see in these figures, in both our complexes, there is a spectra, there is a peak at 260 nanometers, meaning that the aptamer were truly bound to the complex. After that, we needed to measure the enzymatic activity of our complex because it's a very important part of our compounds to have enzymatic activity, so we can use it later. In that graph, you can see the enzymatic activity, firstly of the enzyme, the enzyme, then the aptamer with the enzyme, not our compound, just aptamers and aptamer and enzyme. Then the two compounds that we obtained, the compound with nickel chloride and the compound with EDCNHs. As you can see, we wanted to measure the enzymatic activity of the enzyme in the aptamer to see if the aptamer inhibits the enzymatic activity, and it truly inhibit a little bit, but not in a great amount. And then we measure the enzymatic activity or the compounds. So you can see that the compound with nickel chloride, our main compound of interest, it remains a little bit of a decent amount of enzymatic activity, around 1/3 of the initial enzymatic activity, and the compound with EDCNHs has almost no enzymatic activity, mainly because it affects the active sites of the enzyme. And lastly, we analyze our products using size exclusion chromatography. So there you can see the chromatograph, and you can see the peaks of the aptamers of the FLDH, and it's also, I don't know if it's very visible, but it's a little peak in which you can see the presence of our compound. So our main focus is in the future to modify a little bit of the reaction, so we will obtain our compound in a bigger amount. So as a conclusion, the experiment was successful, and the final goal is to obtain labeled aptamers with both high affinity for the target protein and high enzymatic activity. And thank you for your attention.
Thank you, Du, for this interesting presentation. If there are any questions in online? No, so thank you again. We'll move forward to the next talk, which belongs to Madalina Ghiuță, pristine and modified hollow mesoporous silica spheres as carriers for polyphenols.
Hello, everyone. My name is Madalina. I'm a student in the 4th year, and today I will talk about pristine and modified hollow mesoporous silica spheres as carriers for polyphenols. I chose this topic because hollow structural mesoporous silica spheres are highly attractive in either fundamental research or practical applications due to their excellent mechanical and thermal stability, high biocompatibility, and their unique structural properties. The purpose of this work was to obtain and characterize pure and modified with zinc oxide mesoporous silica spheres, which was used as carriers for a bilberry leaves extract, resulting in composite materials for topical applications. Hollow structural mesoporous silica spheres can be used in various biomedical applications, such as controlled drug delivery based on surface properties, in bone diseases therapy, cancer therapy, as carriers for antibacterial agents for infectious diseases therapy, and in magnetic resonance imaging due to the possibility to link on silica surface a contrast agent. The hollow mesoporous silica spheres can be loaded with antimicrobial compounds, resulting in an antibacterial compound loaded carrier which releases the biological active compounds and kill bacteria strains, preventing this way the biofilm formation. Pristine mesoporous silica spheres were synthesized by sol gel method using tetraethyl orthosilicate and hexadecyl trimethyl ammonium bromide as silica source and structure directing agent respectively. Then a solvothermal treatment was applied to obtain hollow structure mesoporous silica spheres with large and ordered pore mesochannels. In this image, you can see the solvothermal treatment. The obtained materials were characterized by various methods. The small angle XRD pattern of hollow mesoporous silica spheres exhibit one intense peak attributed to 100 Bragg reflection, which is characteristic to MCM41 mesoporous silica, indicating the ordered mesoporous structure. The small angle XRD pattern of zinc modified hollow silica in comparison with that of pristine silica also show an ordered pore mesostructure. And the wide angle XRD pattern of hollow silica modified with zinc oxide suggests that zinc oxide nanoparticles are amorphous or very small. The nitrogen absorption desorption isotherms are characteristic for mesoporous compounds. The textural parameters are determined from the corresponding isotherms, such as specific surface area and total pore volume. They have high values, which means a high porosity, and hence the possibility to accommodate a high amount of organic molecules. The pore size distribution curves were determined from the corresponding isotherms by density functional theory. And as you can see here, the presence of zinc oxide nanoparticles led to a decrease in average pore diameter because they suggested this way the presence of zinc oxide nanoparticles inside the pore mesochannels. The morphology of hollow silica spheres was analyzed by SEM analysis, and they show a very uniform particle size and a well-defined spherical shape when cyclohexane was used as a swelling agent. And in this SEM micrograph, you can see the hollow silica, the fact that silica spheres are hollow. Mesoporous silica spheres were modified with zinc oxide due to its well-known antibacterial properties. The method used was absorption and impregnation using zinc acetate as a zinc source. Based on thermal analysis, they were chosen the condition of the thermal treatment for the decomposition of zinc precursor for both composite samples. These materials were analyzed by scanning electron microscopy, and the images suggest the zinc oxide is found inside the silica spheres in agreement with diffraction data that proved the amorphous nature of nanoparticles. The zinc oxide distribution was evaluated by IDX mapping. As you can see with green in that image, there is a uniform distribution of zinc in the material. TEM investigation also suggests that zinc oxide nanoparticles are inside the hollow silica spheres, and they show that a few of them are crystalline. Polyphenols have been studied intensively in the last few years due to their health benefits like antioxidant, anti-inflammatory, and antibacterial properties. Due to the high porosity of both pristine and modified silica spheres, they can be employed as carriers for natural compound encapsulation. Therefore, it was chosen a polyphenolic extract made by conventional method in 3 stages extraction in water ethanol mixture at 60 degrees Celsius using bilberry leaves. The polyphenolic extract has a high content of polyphenols, flavonoids, and a high radical scavenging activity. The main 2 compounds are chlorogenic acid and rutin hydrate, 2 compounds known for their antibacterial potential. An interesting aspect is the synergy between chlorogenic acid and rutin hydrate present in the extract. The microbial growth inhibition is significantly higher than the one of separate standard compounds found in the extract. The content of encapsulated extract was evaluated by thermal analysis, shown in this image. And the biological evaluation of, of extract supports and extract-loaded materials was carried out on 2 cell lines, fibroblasts and Hackett keratinocytes. As you can see in the graphs, these materials are biocompatible up to 50 micrograms per milliliter dose of treatment for both cell lines. The bilberry leaves extract proved to be highly efficient as an antibacterial agent, showing an inhibition average diameter of 26 millimeters. And furthermore, when zinc oxide was encapsulated, they slightly enhanced the antibacterial potential, showing an inhibition average diameter of 28 millimeters. In conclusion, this work was based on obtaining composite materials from mesoporous silica spheres. Methods of encapsulating zinc oxide nanoparticles in the hollow silica spheres were developed. Both pristine and modified with zinc silica spheres were used as supports for bilberry leaves extract to obtain extract-loaded materials. And finally, the antimicrobial and biological evaluation showed that these materials are suitable for topical applications. That was everything. Thank you for your attention.
Thank you for this complex presentation. If there are any questions? No. So thank you again.
Thank you very much. We invite our next presenter, Catalina Diana Ușurelu, with the work preparation and functionalization of nanocellulose using lactic acid, morphostructural characterization, and emulsifying capacity.
Sorry. Hello, my name is Catalina Ușurelu, and today I will present you the preparation and functionalization of nanocellulose using lactic acid, morphostructural characterization, and its emulsifying capacity. First, I'm going to talk a bit about Pickering emulsions. Pickering emulsions are formulations of 2 immiscible liquids, which are stabilized by fine particles that are irreversibly absorbed at the interface between the 2 immiscible liquids. They have many advantages over the traditional emulsions because they are less toxic and more biocompatible. They are less costly and also they are more sustainable since they don't use the classic surfactants, which are oil-based, but solid particles which are derived from nature, such as montmorillonite, laponite, and chitin. And also it is believed that they have a better stability against coalescence. There are 2 types, of course, of Pickering emulsions: oil in water emulsions and water in oil emulsions. Very attractive as stabilizers for Pickering emulsions can be nanocellulose due to its many advantages, such as its provenience from renewable sources, biodegradability, biocompatibility, its high surface area, as well as the plenitude of hydroxy groups from its surface, which open plenty of possibilities for tailoring its surface properties. However, what really sustains the use of nanocellulose as stabilizers for Pickering emulsions is in fact its amphiphilic nature, since many authors claim that the cellulose crystals present both hydrophobic and hydrophilic edge bands. And also nanocellulose has the capacity to form a 3-dimensional entangled network, when it's dispersed in water, and this network can increase the steric hindrances between the droplets from the emulsion and increase the stability of the emulsion. However, cellulose has the advantage that has an inherent hydrophilic character. So in order to overcome this problem, we can modify its surface chemically in order to increase slightly its surface hydrophobicity, to accentuate its amphiphilic character, and to increase its wettability, especially by the oil phase. The aims of our study was to obtain chemically modified nanocellulose by a chemical treatment with lactic acid, followed by a mechanical treatment by microfluidization. Then to investigate the effect of the chemical and mechanical treatment on the surface chemistry, thermostability, size and the morphology of the obtained nanocellulose fiber, and also to assess the capacity of the obtained nanofibers to act as stabilizers for Pickering emulsions. In what concerns the experimental procedure, the chemically modified nanocellulose was obtained by the esterification of microcrystalline cellulose with hydroxy acid, which is lactic acid, at 100 degrees for 4 hours, followed by microfluidization for 10 cycles. We can see from table 1 that we have tried many recipes. We also attempted to add different amounts of hydrochloric acid, because we know that hydrochloric acid can act as catalyst for the esterification of cellulose with different acids, and also hydrochloric acid is believed to be an adjuvant for the defibrillation of nanocellulose during the microfluidization. We can see that we also prepared the control sample obtained by the treatment of microcrystalline cellulose with hydrochloric acid, since hydrochloric acid is a common acid used for obtaining nanocellulose. The Pickering emulsions were simply obtained by mixing equal volumes of linseed oil and water that contain 0.5 and 1% per weight nanocellulose, followed by ultrasonication. The FTIR spectra showed that regardless of the presence of the hydrochloric acid in the reaction media, all the samples that were obtained by treatment with lactic acid present a new peak at around 1,734 centimeters to -1, which is characteristic to the carbon double oxygen stretching vibration for the from the new ester groups that were formed on the surface of nanocellulose. However, this peak is completely absent in the case of the control sample, the curve in black, due to the absence of the lactic acid from the acid medium. The thermogravimetric analysis showed that the chemical treatment with lactic acid followed by a microfluidization led in fact to an increase in the thermostability of nanocellulose, which can be seen by the increase in the temperature at the maximum degradation rate and in the temperature of the degradation onset. This might be because of the new ester groups generated on the surface of nanocellulose, which are known to be more stable against the thermal degradation. The scanning electron microscopy showed that both the control sample obtained in the presence of hydrochloric acid and the samples obtained in the presence of lactic acid present diameters in the nano range. So we can say that we indeed obtain cellulose nanofibers, and this also means that fraction of the cellulose suspension suffered an advanced defibrillation, since we can see that in the case of lactic acid especially, we can see that the fibers are more individualized, while in the case of the control sample, the first picture, the fibers are more agglomerated, probably due to the hydrogen bonding interactions between the hydroxyl groups from the surface of these fibers. We also studied the rheology of the suspension of the nanocellulose fibers that we obtained, and we saw that all the cellulose nanosuspension present a shear thinning behavior, meaning that their viscosity decreases with increasing the shear rate. This is normal for the suspension of nanocellulose because during the shear stress, the entangled fibril network of cellulose will break and will form cellulose bundles or even individual nanocellulose fibers, which subsequently can align themselves on the direction on the flow and hence the decrease in the viscosity. We can also see that the viscosity of the samples obtained in the presence of lactic acid is higher than the one obtained in the presence of hydrochloric acid alone on the entire frequency range. This might be due to the slightly higher hydrophobicity of the samples obtained in the presence of lactic acid. Which endowed the fibers with a water repellent characteristic, as well as to new interactions between the fibers, maybe of hydrophobic origin. And we can also see that for all samples, regardless of the acid that we use, the storage modulus was higher than the loss modulus, which means that the suspension of the cellulose nanofibers present a gel-like behavior. In what concerns the ability of the prepared cellulose nanofibers to act as stabilizers for Pickering emulsions, we can see from the photos that we took at the initial moment to 1 hour, 3 hours, 1 day, 3 days, and 10 days respectively, that the best results were obtained for NCLA3 and NCLA4, which are the emulsions stabilized by the cellulose nanofibers obtained in the presence of lactic acid and the highest amount of hydrochloric acid as catalyst. Which may mean that they presented the highest degree of esterification, and so the highest hydrophobicity, surface hydrophobicity, which may have permitted a sufficient wetting by both the oil phase and the water phase. The poorest results were obtained for the emulsion stabilized with the control sample, the one obtained only with hydrochloric acid, and for NCLA 0, which were the fibers obtained only with in the presence of lactic acid. We also see from the figure 4 that the creaming index is in fact an indicator of the stability of the emulsions, being directly proportional with the instability of the emulsions. And we can see the high difference between the emulsions stabilized with the fibers obtained by chemical treatment with lactic acid and the fibers obtained by chemical treatment with only hydrochloric acid. The same results remain available when a higher concentration of cellulose nanofibers was present in the emulsions. However, we can see a more accentuated, faster tendency of destabilization. This may mean that excess of nanocellulose was present at the interface between oil and water. And since we all know that nanocellulose has the tendency to self-associate and self-aggregate, this could have led to the coalescence of the droplets in the emulsions and hence the lower stability at higher concentration of nanocellulose. The optical microscopy images are in agreement with the stability studies that I have presented before. We can see that the smallest droplets and the narrower distribution of the size of the droplets was obtained in the last two cases, last two columns, NCLA3 and NCLA4. The same observation remain available at concentration of cellulose in the emulsions of 1%. In conclusions, we obtain chemically modified cellulose nanofibers by treatment with a weak acid, lactic acid, followed by microfluidization. We have seen that the chemical treatment with lactic acid led to the simultaneous chemical modification of lactic acid, to the simultaneous esterification of cellulose with lactic acid, and also aided in the defibrillation of cellulose during the microfluidization step. As shown by the thermogravimetric analysis, the modification with lactic acid led in fact to an increase in the thermostability of nanocellulose, and we also have seen that the cellulose that was chemically modified with lactic acid acted as a great stabilizers for Pickering emulsions, while the control sample obtained with hydrochloric acid, which is a strong acid, did not perform as well in this aspect. Thank you for your attention.
Thank you very much, Catalina. If there are any questions from the room? Okay, Professor Sârbu.
All the presenters were in a very hurry to speak fast. They could not press during their speech. Sometimes I think that they will collapse. So exercise your way of speaking so to make time also to inhale air from time to time. Otherwise, congratulations. But
thank you.
All of you did the same.
Probably they try to present as many results as possible in the short time allocated, yes.
Okay, you had a very nice presentation.
Thank you.
With a lot of data. My question is, did you measure the diameter and the length of the nanofibers? And if there are differences between hydrochloric acid and lactic acid?
Not yet, but I have made some measurements on the diameter, if you have seen. There is a small difference between the ones obtained in the presence of hydrochloric acid. They have a slightly higher diameter, but I think some images do not help us to measure the length of the particles. Because you can see that they are agglomerated, they are overlapping, and we can measure the length of the fibers. Maybe by other method, sometimes. Why it is important to measure the length?
Composite. It's the properties of the composite, because you will do later composites with this, I think.
No, I use them as stabilizers for Pickering emulsions. Yes.
You don't use later this, it's only theory?
No, I do Pickering emulsions with them. I use them as stabilizers.
No, I don't understand which is the field of application of your material.
Well, they can be used in cosmetics as creams, as ointments, because linseed oil has antimicrobial and also antioxidant properties.
Okay, but in cosmetics, for instance, it's important the length, because resistance of the coverage.
Yes,
it depends of the length.
Yes, thank you, thank you.
And not only that, but everything that comes in contact with human skin should not be in the nanometer range, because we go to other discussions. Okay. If there are any other questions from the room or from the online participants? If not, as we are a little ahead of schedule, which is surprising, I would like to see if our colleagues from yesterday online poster presentation are online right now. So Vasile Guțanu, Oleg Petuhov, Alina Mariela Ipate, Gabriela Lisa, and Maria Botnaru, if you are online. It seems like they are not. Irina Georgiana Munteanu and Constantina Petrei from Dunărea de Jos University. No, or Sorin Claudiu Linici, Gabriela Băisan, Alin Stoica, and Radu Mărarcă. No, it seems that they are not online. We will make another try after the break. So I will invite you for a 15 minutes break, a coffee break, and to continue our discussions outside. Thank you.
So, good afternoon. My name is Nicola Bogdan, as the chairperson said. Today I'm going to be talking to you about catalytic micropumps made of urease. Moreover, then I wrote in the abstract about catalytic micropumps made of glucose oxidase. First up, I want to talk about what are these micropumps, these enzyme micropumps. These are immobilized disc-shaped patches of enzyme on a surface that once in a solution of their substrate, they generate these substrate and product concentration gradients that generate in the solution two types of flows, soluble buoyancy driven flows, and also these soluble buoyancy driven flows take place in the bulk volume of the solution, and also an osmotic flow that takes place at the interface of the enzyme at the wall. Now, to give you some context on why or what are the uses of these devices, they can serve as drug delivery devices, or by case as biosensors. In this example from the literature, they had a hydrogel that was decorated with these enzyme micropumps. I believe that it was glucose oxidase, the enzyme, and in the hydrogel was entrapped cargo molecule, in this case insulin, and what they observed was that when the substrate, in this case glucose, was added in the solution, the release of the insulin was increasing in a dependent manner of the glucose concentration. And as a second use, and a very promising one, is as mixing or pumping liquid in confined spaces, such as microfluidic channels. What you can see in the figure is just a simulation, a mathematical simulation, but you can see that arranged in such an array, these enzyme micropumps could transport the liquid from one part of the microchannel to the other. Next up, I'm gonna be breaking down these two flows that I talked about. The first one is the bulk flows, the soluble buoyancy driven flows. What you can see in these movies on the center, I believe it's too bright to see in the room, but bear with me. What you see in the center of the movies is the micropumps, and it is notable that first of all, we observe this with an optical microscope, and we set the focus of these experiments at 170 and 850 microns above these micropumps, and with the help of these so-called tracer particles, which are 2 microns polystyrene spheres in this case, we observe the direction of these flows. In this case, at 170 microns above the surface, the tracer particles are being pushed away, and at 850, they are moving towards the micropump. This tracking, this particle tracking, also help us with determining the flow speeds. As you can see, the values of these speeds was not dependent on the substrate concentration, and it was pretty similar in both cases at 170 and 850 height above. So this is a schematic explanation of what I just explained. This is the urease micropump. Oh, this is the urease micropump in urea solution. In the movies you saw was 8 millimolar urea. The diameter of the micropump was about 900 microns, and these are the flows that are generated in the you may be someone may or to further add proof to this theory of the that these flows are generated due to the concentration gradients of the product and the substrate. We conducted experiments in which the parameters were the same, except we turned the whole setup upside down. So the micropump this time was up above. And so this is what we obtained. As we expected, the flows reversed. So at 170 distance, this time below, the tracer particles were moving towards instead of being pushed away towards the micropump. And at 850 below, the tracer particles were being pushed away. So again, this is the schematic explanation. The same pump, this time turned upside down, that generates this time flow, a convective flow that moves inward. Like next up, I'll talk about the osmotic flows, also called the surface flows. This is how they manifest. They push away the tracer particles. This time we use some heavier particles that land on the surface, and under the force of the flows, they slide on the surface. What we obtain is the so-called depletion zone, a zone depleted of these tracer particles. And this depletion zone can be correlated with many things, as you can see. As we expected, it was we could correlate it with the pump diameter, and as we expected, and as you can see on the left, on the graph on the left, a smaller micro pump was producing a smaller depletion zone and slower in time. Now, again, the schematic explanation. These micro pump, this time the glucose oxidase micro pump in a solution of glucose produces these osmotic flows at the surface that push away the tracer particles, and for let's say 260 microns in diameter glucose oxidase micropump, we obtain a depletion zone with about 300 microns. One of our recent studies, I'm sorry you cannot really see on this screen what exactly is happening. In one of our recent studies, we wanted to see the impact of the some parameters on these osmotic flows. In this case, the impact of the surface properties. On the left movie, you should have been able to see that tracer particles were moving towards the glucose oxidase micropump on a polystyrene surface. And on the right movie, you should have been able to see that particles were being pushed away on a BSA bovine albumin surface, a coated surface. So as conclusions, I forgot to say that these micropumps that we obtained are some of the smallest described in the literature. These produce robust and measurable bulk flows and osmotic flows. And these are actually promising devices for what I've said, mixing, pumping, and why not as biosensors. In the end, I want to acknowledge my supervisor and my mentor, Sylvester Gaspar, and to the professor that helps us with the mathematical models, which is the theory that stands behind all of these experiments. And I want to thank you for the attention.
Thank you too for the presentation. If there are any questions, I would like to ask you if you publish already your data.
No, we didn't. We are on the brink of publishing it.
Thank you again.
Thank you.
Thank you very much. Our next presentation, I think it's from online. Diana Ioana Buliga, optical and tinctorial properties of some natural dyes embedded in hybrid silica coatings.
Yes, good evening. Can you hear me?
Diana, if you're online.
Yes, I think I'm having some audio issues. Can you hear me?
Yes, we can hear you, but very... And please share your screen.
Yes, just 1 second. Can you confirm that you are seeing the presentation?
Yes, please go full screen.
Yes, I'm trying to. Is it okay now?
Yes, it is okay.
Okay, thank you.
Proceed.
I think I'm having a little bit of delay with the audio, but I think we should be fine now.
Yes, we can see the presentation, so.
Okay, thank you. Good evening. Thank you for your introduction. Today's presentation is about optical and tinctorial properties of some natural dyes embedded in silica coatings. The natural dyes used in the current study are alizarin and phycocyanin. To improve their light fastness, hybrid silica coatings were used, and the resistance to light was carried out with Xenotest 150S+. Spirulina plantensis is a blue-green alga with important functional value and possible future as a commercial blue textile dye. Phycocyanin is a pigment protein complex from the phycobiliprotein family, which acts as an accessory pigment to chlorophyll. Phycocyanobylin, which is actually the reason why the blue hue is present, is the tetrapyrrole chromophore present in the phycocyanin. Alizarin is a known reddish natural dye extracted from the roots of Rubia tinctorium or a more commonly known madder. It was used from ancient times as paint or as a mean to dye clothes, and today it is also utilized to identify osteocytes, which is a procedure extremely important for osteoporosis investigations and tracking. The two natural dyes, alizarin and phycocyanin, were extracted using conventional and unconventional extraction methods with their corresponding parameters and concentration determinations. The employed hybrid silica coatings are based on tetraethyl orthosilicate and phenyl triethoxysilan, together with tetrahydrofuran, ethanol, citric acid, or hydrochloric acid. The goal of this research was to determine a method of color stabilization. Regarding light fastness, several solutions were prepared and used to dye a cotton blend fabric. As seen here, the textile was colored either from the natural dye dissolved in one of the two solvents or with a stabilizing agent that was used to do a second layer over the already dyed cotton blend. A similar approach was used for alizarin, but the two starting points here were of either natural extraction or of synthetic origin. Although many trials were performed, I chose to only depict the nanosol synthesis, where the first step was to put in contact the natural or the synthetic dye and the sodium alginate solution, followed by the capsule precursors, which were actually, in our case, theos and phenyl tes. The textile material was dyed using a horizontal laboratory padding mangle, as shown here. After drying the dyed fabric, the samples were subjected to a light fastness testing aided by the Xenotest 150S+. The light exposure was maintained for 180 minutes in total, with increments of 30 minutes each. Sorry. The results of the Xenotest were observed regarding the CLAB coordinates. As we can see here, for phycocyanin, delta E, which is actually the observed hue difference between time 0 and 180 minutes, has values of over 14 in almost all cases, which means a natural dye has a poor light fastness. Here, the differences can be better observed. Results with almost 16% smaller delta E were obtained when the 2% alginate solution was utilized and not the buffer solution. However, the best outcomes were obtained when nanosol was impregnated on the already dyed fabric for both cases. We can see here that for the phosphate buffer, an improvement with almost 26% resulted for sample F6 as compared to sample F1. And for the sodium alginate solution, a 9% better light fastness was obtained for sample F5 as compared to sample F2. In the case of alizarin, the light fastness has better results than for the other natural dye, with the lowest delta E being recorded for samples A2 and A9. It is important to notice that almost all samples here have a hue difference value below 4, which implies good resistance to light. The extracted dye as such, which means that either synthesized or extracted, but without any nanosol or any other stabilizing agent added, exhibited the best light fastness. And this is valid for, of course, only the used methods. The synthetic ones, showing already an improvement with almost 40% better light fastness as compared with sample A1, on the other hand, excuse me. So as I was saying, for the synthesized alizarin, the synthetic one, although we have better light fastness already than in the case of the naturally extracted one, adding nanosol improved the light fastness even more, with almost 15% than when compared to sample A2. To sum up, natural dyes have always fascinated mankind, but they have been known to be easily degradable at almost all the environmental factors. So it is very important that if you want to pursue this a new way of doing chemistry that is now trending, let's say, to stabilize these dyes and to make sure that they can be made commercially available. So for our case, when using phycocyanin and alizarin, we can say that although phycocyanin exhibits an overall poor light fastness and lower resistance than alizarin, both dyes showed improvement when nanosol was impregnated on a second layer. So this might mean that if we continue the research, which we all intend to do, we might be able to come up with a commercial dye that has a better view for the environment in the future. We have here some selective bibliography. Thank you very much for your attention, and I'm so sorry for all the coughing, but I'm just recovering after COVID and after the flu. So thank you.
Thank you very much, Diana. Very nice results, and with practical application, a very good example of collaboration between two groups, one from Politehnica University of Bucharest and our colleagues from ICECHIM. If anyone in the room have any questions or from online? If not, thank you once again, and I hope to see you again healthy in our colleagues' laboratory.
Thank you. Thank you very much.
Thank you. For the next presentation, I invite Mr. Radu Albulescu to present us the work entitled Proteogenomic Tools in the Assessment of Pharmacological Effects of Natural Compounds.
The opportunity to be present here. What we have here is a very nice study. If you want, okay, let's go. So we already are aware of proteomics as one of the main tools in deciphering the mechanism of life and also of disease. Proteomics means the study of all protein components in a certain medium, that is a total organism or a fluid of an organism, a cell, and so forth. So what is characteristic? So the first omics tools that were developed were genomics and proteomics, and what is characteristic is the much larger complexity of the proteome compared with the genome. We have about 1 million proteins in humans, and these proteins are produced of about 30 to 40,000 genes. So I illustrate first the central dogma of biology, that the proteins are the key. We start from chromosomes. Each chromosome has a subset of genes. There we have DNA double helixes. From the DNA double helixes, we go then to transcript that suffer RNA splicing. So the elements, the programming elements are joined together, and from RNA we go to translation into proteins. So it's possible that from one double helix to obtain several different transcripts, and so to program different proteins from the same gene source. We have 20 to 30. I said the information goes beyond to about 40,000. We have 100,000 of mRNAs, and then we go to proteins, and the same individual can achieve different sets of proteins during his life. So we have about 1 million proteins in our body. So DNAs and RNAs are in fact only templates for building proteins. They are very important, but the protein that are responsible for specific activities in the body are the proteins. And they may represent better tools for understanding the functioning of our body, either in health or in disease. So speaking of proteomics investigation, there are many tools already available here. So the most, let's say, comprehensive and performant for the level is represented by mass spectrometry. And is largely used nowadays in the study of proteomics, even if sometimes we still used for sequencing the old Edmond sequencing as to identify protein sequence. Even if it's very performant, mass spectrometry has still some limitation. We can have also a second stage proteomics, which is based by using methods of identification and or quantitation of the proteins, generally based on the use of specific reagents, mainly labeled antibodies. And we have several distinct technology to make then quantification. We have cell-D, MALDI devices. We have Luminex, XMAP. We have protein microarray, BIACOR instruments. All of this acting in making protein identification. So what the problem is, one of the major problems is that in the proteome, about 90 to 95% of the total protein content is represented by a few major proteins. And most often, the interesting proteins for physiology and for pathology are present in very low amounts. So what can we do to detect them? Some of them can be detected, others are still very problematic. Here is a map of protein abundance in human liquid, and you see the major protein representing about 90% of the protein. They are in large amounts. They can be easily identified, but they are not often interesting as the protein present in the right down square. So we want to detect those proteins because those proteins are generally representing signature of disease, so biomarkers or signature of responses to several conditions. So we have solutions, but the solution to amplify this concentration are most often difficult to achieve, not only because we need to concentrate those proteins, but because we would need large amounts of sample. So how much blood can I collect from a subject to concentrate the protein and to obtain good results? And how often can I collect the blood to make such analysis? So the solution are, but are not very popular among patients, among doctors. So we must find something else. So the landscape of this field changed very rapidly since 2010, because someone had the idea to couple the detection on antibodies with single-stranded DNA and to provide from a non-amplifier detection an amplifier method of detection of protein. So we can 10 to 50 times amplify the signal of a protein by such technology, and we can achieve then the detection of small concentration proteins without need of large amount of original sample. So the first commercially available such technology was the pro-quantum technology. It was using some DNA fragments coupled to antibodies, and most of the applications are in the diagnostics field, but we want to move towards the non-clinical applications. And it's very important that such a system can be applied on any real-time PCR. It doesn't need a specialized equipment. Now, since 2021, emerged a second technology. We will not have examples of this in the presentation. This is the commercially branded as O-link technology. This requires specialized equipment to achieve it, while the pro-quantum technology could examine one protein at a time in a sample. This technology is a multiplex technology which allows 40 to 80 analytes to be detected simultaneously. And moreover, the sample volume is required for the application is 5 to 10 microliters for the whole set of the 80 analytes. So it's a very, very nice and promising technology. And we can use this technology both on extracellular proteins and also on intracellular proteins like those that are involved in signal transduction. And we can see if these proteins are differently activated, for instance, by phosphorylation. So we were here. We again, it's a bit of remembering that nucleic acid represents a pool of basic genetic information. The proteins represent the support that allows the achievement of functional characteristic of an organism. And this means that the proteins present in a cell, a tissue culture, an organ, or an entire organism are making possible to evaluate the potential modification induced by, for instance, physiological or pathological condition. We have some advantages of the new proteomics, proteogenomics technology towards the classical one is that in classical proteomics, we cannot have amplification of the signal. Usually, the measured concentration of a protein is between 10 and, let's say, 50% of the real concentration of that protein. And so in many situations, the concentration of the effective concentration is below the limit of detection. But using the proteogenomic approach, we can enhance the signal and obtain much better results. Here we have some comparison, what we can do for a cytokine using the pro-quantum immunoassay compared with the classical assay. And you see the broader dynamic range achieved by pro-quantum. So we can quantify in smaller volume and even at lower concentrations. And the experimental part of the study was achieved using two different technologies, which we used for the first time in Romania. One model is the use of the EpiSkin model to evaluate the potential irritability and corrosivity. And the second one was the application of pro-quantum technology to detect cytokines.
Sorry, can you please prepare to close in 2 or 3 minutes?
Yes. So the results here, we have the testing of toxicity by EpiSkin model, followed by, and you see we had several samples. And after that, we went with the same sample in measuring the concentration for 4 distinct cytokines. And you see that the level of each of them in various experimental conditions all the time the exposure was to CBDA at 2 concentration, and we measured the concentration of the molecules in the supernatants of the culture and also in the lysate of the cell. And we go to the conclusion. So this approach affords a major advantage by using the amplification, and it has very good precision and sensitivity. It allows us to investigate pharmacologically and when we use in vitro system in very low volumes, like we can use even not only 96 well plates, but I think 392 wells could be used for the exposure experiments, and the amount of sample is enough to achieve measurement. And we have also the possibility to perform some in vivo studies as well, since the volume of samples collected from an animal subject could be about 100 microliters at an assay, and this is very compliant with the 3 Rs initiative. So that was briefly what we did. We obtained funding from this, and the most interesting slide is see here what different scientists are looking at in pharmacology.
Thank you too for this nice presentation on protein detection tools. And if we have any questions in the room or from online. If not, thank you again.
Thank you. We now invite our colleagues from the National Institute for Optoelectronics in 2000, Iulian Pană, Anca Constantin Părău, Mihaela Dinu, Diana Maria Vrânceanu, Cătălin Vițelaru, and Alina Vlădescu Dragomir to present the work Chromium Silica Carbonitride Coatings with Enhanced Mechanical and Anti-Corrosive Performance for Woodworking Industry. I think our colleague is online, so please.
Good afternoon, everyone. Thank you for your kind introduction. So today, I'm going to have a presentation that is not only about possibilities, but quantifiable results. So in the next few minutes, I'm going to present some insights into woodworking industry challenges. So the main question that we have to answer is, why wood is so important? So we know that wood is used in various industries, starting from construction up to furniture manufacturing. So the efficiency of wood processing is heavily relying on the on the tools that we use for this. So this brings us to the importance of wear resistance materials for woodworking industry. So we all know that metallic alloys are strong and versatile, but when it comes about applications that require wear and abrasion, their integrity can be, and durability and longevity can be be compromised when they are exposed to harsh environments and repetitive mechanical stresses. So this is when hard coating enters into the picture. So basically, hard coatings protect the metallic alloys from and enhance their resistance to wear, corrosion, and other forms of degradation by preserving the properties of metallic alloys, extending their lifespan and performance. So why are we particularly interested in chromium silicon carbonitride coatings? The response is quite simple, because due to their unique combination of properties. So our main goals in this research are twofold. So first, we want to study the influence of nitrogen to carbon ratio to within the chromium silicon carbonitride coatings, and to study the influence of carbon to iron ratio between the substrates. So we want to know how they outperform the conventional chromium nitride coatings and chromium carbon nitride coatings, because several studies have already indicated that the silicon nitride amorphous phases decrease the wear. Oh, I'm sorry. Decrease the wear and also increase in durability. So to achieve our objectives, we employed reactive cathodic arc deposition in order to obtain chromium silicon carbonitride coating. So we have used 3 different steel substrates with different iron to carbon ratios, and we have obtained 3 different carbonitride coatings under 3 different reactive gas flows that we can see here. It's 30 to 70, 50 to 50, and 70 to 70. So we have include some silicon into our layers by using composite targets formed by chromium silicon. So first, we have studied the morphology and the composition of the substrate, and we have seen that we have different iron to carbon ratios, which is good because they actually influence a lot the physical properties of the substrate. And to explore the morphology further, scanning electron microscopy and energy dispersive X-ray spectrometry was employed in order to reveal the distribution of the constituent elements. And we have seen that all the mainly carbon, iron, and alloying elements are uniformly distributed, with the mention that in the case of still one substrate, the EDS mapping analysis revealed the presence of carbon-rich phases, which are known to strengthen the coating matrix and enhance their adhesion. So the XRD analysis of the steel substrate revealed that iron is a common element that is shared by all these substrates. One observation was in the case of steel one substrate. We have seen that besides the cubic alpha iron phase, in the case which is common for all the substrates, we have seen that this is particularly for the S1 substrate. So the monoclinic iron carbide phase which is here showed by orange color, means that this is due to their heat treatment of the steel substrate. So the SEM analysis of our coating showed what we intended to do, that carbon content is increasing for all of our coatings, which is here represented by the green color, while the nitrogen is decreasing among our substrates. That proves that we have a tunable carbon to nitrogen ratio. Also, this tunable carbon to nitrogen ratio is increasing as we change the reactive gas flows during the deposition. So also we have seen here that with red, that silicon is present also in the coatings. This being a result of the chromium silicon cathode that we have used during the cathodic arc evaporation process. So the adhesion was evaluated via scratch test. So the aim was to pinpoint the critical force where the delamination of the layer occurs. So we have observed that the coating deposited on steel 1 exhibited on a good adhesion. I would say an outstanding adhesion at with critical force values that are ranging between 17 and 18 newtons. So we can say that we can conclude that carbon-rich phases that we have observed during the morphology evaluation help to form strong bonds between our coatings and the steel substrate. So moving on the right side to the roughness, we have polished our steel substrate for to roughness is up to 15 nanometers. So we have observed in the case of all coatings that the roughness is increasing. This is a particular characteristics is a common occurrence in cathodic arc evaporation process. So we notice that the type of alloy, it is not important here because and it is not influencing the roughness of the coating. So moving forward, the tribological test revealed that our coatings outperform all the steel substrate. The steel substrates are represented here with the black color, and we see that the friction coefficient is for the whole coating are below the black curve. So, but the best adhesion was for the layer with the green color. This is with 50% of acetylene obtained with a reactive gas flow of 50 standard cubic centimeters per minute of acetylene and 50 SCCM for nitrogen. So the other two coatings exhibited an unstable behavior. So and the wear was also the lowest wear rate was you can see here below in the right, that was obtained also for the middle the coating in the middle in this representation. So furthermore, we have evaluated the corrosion resistance of the coatings via potential dynamic polarization method by using distilled water and sand at the room temperature. And we have seen that as a general remark, the coatings improve the corrosion resistance of the steel surface, which was expected also. But all the layers deposited on the steel one substrate demonstrated the most electro positive value of the corrosion potential, indicated a relatively better corrosion resistance than S2 or S3. So once more, the chromium silicon carbonitride with C50 and N50 on steel one substrate demonstrated the best corrosion resistance. So even if the corrosion current density was found to be lowest for the next layer on the steel one substrate, we think that the chromium silicon carbonitride with C50 and N50 was the best for the other study. So transitioning from lab scenarios to real life testing, we have chosen to in-depth testing our chrome silicon carbonitride C50 and N50, and we have covered some planning and cutting tools in a company, and we have put some different wood to be cutted, and we have even if we have using aggressive and easy planning, we have obtained encouraging results by doubling the quantity of wood. We can see here the uncoated tools as they are provided by the commercial suppliers versus our tools that were coated with chromium silicon carbonitride tool. So we have seen here that our coatings almost double the service life of the tools. So as the conclusion, so we have obtained tailored the carbon to nitrogen ratios of chromium silicon carbonitride, and we have seen that the corrosion resistance is improved as compared to the steel. We have obtained low friction and low wear resistance for woodworking applications. So adhesion is improved if the carbon content is increased in the carbon silicon carbonitride layers composition. So practically wood machining demonstrated the benefit of using this type of coating in order to change or replace or to have the tool more to cut more with a two-factor. Yeah. So in the end, I would like to acknowledge the financial support and to thank you everybody for your attention.
Thank you very much, Julian. Congratulations to you and to your very strong research group. We know your activity. So if we have any questions from the on-site participants or from online? If not, we thank you once again for your presentations.
Thank you very much.
To all our participants, I would like to tell that this evening, especially for the one that presented their posters today, tonight they will receive the posters selected for tomorrow. They will receive the announcement, so I will ask them to confirm their presentation until tomorrow morning at 9 a.m. because we have to make our schedule. And this concludes our sessions for tonight. I invite you to also visit the posters in the ground lobby. And we will see you tomorrow at 10 a.m. Thank you very much.